Remember that in order to copy a cell, you need to copy all the DNA first in order to share it out evenly between the two daughter cells. Now this happens in the S phase of interphase in the cell cycle. This lesson is all about exactly how DNA copies itself during DNA replication. Now the first step is to unzip the double helix, okay, to break the hydrogen bonds joining the two polynucleotide strands. And to do this you need an enzyme which is called DNA Helicase. Now once it is unzipped, free nucleotides can come in and match up via complementary base pairing, forming hydrogen bonds between the old base and the new base, so A's with T's and C's with G's. And now another enzyme is involved in this process, and this enzyme is called DNA Polymerase. So once DNA Polymerase has been along and lined up the new bases with the template strand on the old DNA, then you need DNA Ligase, a third enzyme, to come along and bind them all together, making the new strand complete by making phosphodiester bonds. Now this happens on both of the strands. So when you unwind the original double helix, you can replicate both sides of it. And this is called semi-conservative replication because it's half of the original DNA is conserved, is kept the same, semi-conservative. So on the new copies of the DNA, on each copy you've got one strand which is the original DNA and then the new strand that has been formed along with it. So it's semi-conservative replication. And this was discovered by some great scientists, Messelson and Stahl, who did some experiments using different isotopes of nitrogen that you can then find in the bases in order to almost track how much of the old DNA was combining with new DNA and therefore realizing that it was semi-conservative.